Try now.
Hi, can everyone hear me okay?
Yes.
Awesome. Thank you for bearing with the technical difficulties. My name is Virj Khan, and it's wonderful to be here, and I appreciate you all for staying and listening to my talk. So as Pierre mentioned, I'm a designer, I'm an engineer, I'm a media artist, and recently a startup founder entrepreneur. My work targets and focuses on building interface technologies that bridge humans with worlds of information that are either too abstract and incomprehensible or too subtle and imperceptible. And my goal is to reshape human relationships with the environment and each other by investigating the interconnections that are around us. Currently, I run a public benefit company called Primitives, where we work on engineering functional, biodegradable, and intelligent materials to address environmental issues such as greenhouse gas emissions from food waste and plastics pollution, as well as ocean conservation. And my journey began as an academic researcher in the field of human-computer interaction, where I worked on 3D robotic interfaces, shape displays, and wearable technology, and designed new ways to interface with information that, in order to sense and display, create user interfaces and user interactions. One body of information that has captured my interest because they've been largely limited for interface applications is in the areas of food, climate, and the human body, which seem to be incompatible with the current tools for interface design, which is primarily electronics and software. And that's because signals in the biological and chemical world are mediated through a flow of fluids and communicate through chemical codes. Researchers in chemical and biological engineering fields often use chemical mechanisms, reactions, and expensive machinery for sensing and analysis, but they often don't cater to the interests of creative applications or human readable outputs. Technologies and tools for chemical and biological sensing are for human interaction are primarily tied to human readability. So that is the core motivation of my research, is to offer capabilities to create user interactions with these systems that provide this. Today, I'm going to be going through and sharing my body of work in the field of biodesign and a practice that I call molecular design interactions, which represents a culmination of projects I've done to explore creative applications for chemical and biological engineering tools, where I explore projects that examine information landscapes in the body, within our food, and within our environmental systems. So the first project I'm going to be sharing is organic primitives. It's a project that is looking into how do we create material-based interfaces. So a tool, in response to the lack of tools that are out there. So these are a set of stimuli-responsive biomaterials that can change odor, change color, and change shape, and to facilitate interactions with fluid-based systems. And I envision that these materials can take in inputs. In this case, it's pH, but also a variety of other inputs such as dioxins, glucose concentrations, proteins, and introduce them into human readable information outputs such as color, taste, smell, and shape, which are the perceptual building blocks for humans. And the way that they work is primarily there is a dopant, which is integrated into a substrate and a crosslinker, and that material is then applied to an object. We created 3 materials as a result of this work. One that changes color, produces a variety of colors based on pH, and the second material releases odor and can controllably switch the odor on and off based on pH. And we ran studies to assess their human readability of these materials. And this last one changes shape and produces a variety of different angles of shape output. And this allows me to then create a body and a library of materials that then HCI researchers and designers building interfaces can use. And this allows a layer of material primitives, which is just the base materials that take input chemical input and change into color, produce outputs in odor, and produce output in shape. And multimodal composites, which allow you to make a shape and color changing material, shape, color, and odor changing material, and et cetera. And then also integrate them into different forms, such as fibers, films, and three-dimensional forms in resins. And then finally, they can be integrated into provide more additional advanced computing capabilities, such as integrating them into fluidic channels to create devices that can mix different fluids together, titrate solutions, and tell time. As a result of some of this work, I was exploring different application spaces on how do we integrate these materials as a medium to build interfaces with. And so this is an example of it utilized for plants, transforming the plant into a display. Now you can make the plant bleed red. Or an apple that tells you it's been contaminated during transport by breaking out into red spots. An umbrella that bleeds in the presence of acid rain to tell you, ouch, the sky is hurting me. So this is an example of how an application of an apple is transformed into a sensor and display. So when a user washes the apple, it makes contact with the material's organic primitive. And this film is tattooed or integrated as a coating onto the edible surface of the apple, which then generates an output pattern that is determined by a designer. In this case, it's red spots, which draw from the toxicity represented from spotted mushrooms that signify the apple is contaminated. So I draw upon these sort of biosemiotics as interaction metaphors in the case that I just showed you around the spotted apple, but also with the umbrella, the sight of blood, which is very innate actually to, as a biological communication, the sight of blood translating to pain. And other applications, which I haven't shown, which is a shirt that refreshes to new designs when a perfume is applied, or a fork that wilts to tell you to stop eating. So this body of materials allows me to now integrate into different applications, such as food, climate, and plants as a new medium. So that's organic primitives. I will tell you more about where it's at, the project is at now, later on in the talk. Now I'm going to show you a brief project that I was doing in collaboration with Katia Vega, where we were exploring this question around identity. This was a part of a music video that is being produced. This is a small piece of the project that I'm sharing today. And these are called microbial portraits. And the big question that I was pondering and asking in this work was around identity. Who am I if beneath the surface and the face that I know as my own is colonized by a constellation of microbial communities? And so this is a time lapse of a microbial portrait that's captured after human faces were plated on growth media. And the result is a snapshot of a home to millions of microorganisms called the skin microbiota. And the face is not just the self, but also millions of microorganisms. Inhabitants include the corynebacteria and the propionobacteria and the streptococci. Oh, sorry about that. Progressing through slides too quickly. And so essentially the microbial portraits sort of explore and reveal another layer of identity, one that's rendered by a biodiverse ecology that each one of us gives life to. So this is a very brief project. Next project is an exploration of also the human body, but examining the microbes that are throughout to the body instead of just on the face. So this is Microbiota Beats. This is a project done in collaboration with David Kong, Andrew Guo, Yi Xiao, Shannon, and etc. from EMW Street Bio. Biota Beats is a microbial record player that translates growth into new auditory media where microorganisms in the human body is sonified into music. Historically, music has been experienced and stored in a variety of forms. Take the vinyl record, for example, where sound is represented by small engravings scribed onto a material. These engravings are static representations. But utilizing biology, we can create a new medium for music that is dynamic and allows us to experience a record that grows and evolves over time. So the system encompasses three main elements: a retrofitted record player converted into a incubator with a microcontroller and heating element. The second is a custom Petri dish we call a biota record, which structures the growth of the organisms for the camera for detection. The camera captures the images which software algorithm detects and sonifies its growth. In our record, our biota record, we culture microorganisms from different parts of the human body in order to capture a new understanding of human microbiota, specifically the mouth, feet, armpit, genitalia, and button, because these represent diverse, distinct communities. The user experience of our microbial record is drawn from current turntable DJ interactions, which include mixing, sequencing, scratching. This first example is a plate where by swabbing different parts of the human body, then you apply it onto the plate. Once it's plated, it is placed in an incubation chamber. Over the course of several hours, days, growth occurs. The resulting record is one where you can swap a biota record and listen to it, which represents a snapshot and a state that is then sonified. Since a biota record is a living and growing soundscape, each record also embodies a range of sounds over time as the colonies grow. Alternative music can also be generated by the growth over time as opposed to sonifying a single state in the record. So this is an example of some of the sounds I'm going to be showing. So I'm going to stop share, flop to the website real quick so I can show you what it sounds like. So can everyone hear this? What now? Let me try one more time. Can you hear? Okay, that's okay. Well, if you guys are curious, you can go on the biotabeats.org website to listen to the sounds. So no problem. So back to the presentation. Can you hear me? Okay. So in terms of how the data is, the sonification happens, each area in the biota record represents a different area of the body for which microbes were incubated. The camera system captures an image. The image processing algorithm vectorizes into 5 areas, detects the clusters where the colonies of the organisms have grown, and Cartesian coordinates are then assigned to each colony. Musical notes are mapped based on the cluster location, density, and distance from each other. So that's Biota Beats. Again, if you're interested in listening to sounds, go to biobeats.org. Next project is exploring health information using tattoos. So this is a project called Dermal Bis in collaboration with Katia Vega, Nan Zhang, Ali Yedison, Nick Berry, and Shin Liu. We published this work in collaboration with Harvard Medical School. and won a Sci-Fi No More award at South by Southwest. So this project is it's called Dermal Abyss. It's a biointerface proof of concept where tattoos were used as information displays for showing health data, where the body surface is rendered as an interactive display. Traditional tattoo inks are replaced with biosensors whose colors change in response to variations in interstitial fluid. It blends the advances of biotechnology with traditional methods in tattoo art. We investigated 4 biosensors reacting to 3 pieces of biochemical information in bodily fluid to change color. The pH changes between purple and pink, glucose shifts between blue and brown, sodium and a second pH sensor fluoresces at a high intensity under UV light. Dermal Abyss creates a direct access to compartments in the body and reflects the inner metabolic processes in the shape of a tattoo. And it could be used for applications in continuous monitoring, such as medical diagnostics, quantified self, and data encoding in the body. Preliminary evaluation was done as part of our research in an ex vivo pigskin model, and several injections were done in order to understand the visibility, functionality, and of the biosensors in skin. And so this is the project around interfacing with the body and specifically with our health. And this is the sort of my latest endeavor, which focuses on packaging materials and how they mediate our interactions with food and also the larger economic supply chain and environmental system. I view packaging surface as an interface with the ecological food chain and also the international supply chain. And eventually, I hope to bring some of this work to reduce greenhouse gas emissions. And so as this is part of the work that we're doing at Primitive Biodesign, where I'm the CEO, and we make functional, intelligent, biodegradable materials. And this is some of the work that's translated from the work that you saw earlier of the project called Organic Primitives. Specifically now we're focusing on flexible films to create and also sense environmental factors and food deterioration. Using the materials, we are creating a compostable packaging that reduces waste by sensing and displaying food quality. So you know when food is still good to eat. So this is an example of an application where leftovers can then tell you how old it is, how long it's been left in the fridge. In this case, 5 days. Fish that then communicates how long it's been left in the counter for while it's defrosting. And packaging that sends a spillage to let you know when it's no longer safe. So people don't have to rely on guesswork or sniff tests any longer. And the idea is if the whole package must go, the whole unit can then be composted without needing to depackage. And this allows us to reduce food waste and plastics pollution because the two issues are linked in that spoiled food is often encased in plastic, which ends in landfill releasing methane, which is more potent than CO2, 86 times more potent over 20 years. So this compostable packaging, the goal is to divert that waste from landfills to the compost streams and also to consumption, letting people know that it's still good. We decided to target flexible films because and when you think about flexible films, it's those chip bags, it's those plastic bags that are very thin, very lightweight, and usually typically have a multi-layered construction. And this is the area that of focus because oftentimes these films currently do not have a path to recycling. They all end up in landfills and end up also in the oceans because of how light they are. So this is our aim. Our aim is to integrate multiple layers together, including our sensors, different functional properties into one biodegradable film system that utilizes biocomponents from nature, sourced from carbon neutral crops like macroalgae and also agricultural waste. And we're targeting greenhouse gas reductions at every stage of the product life cycle. And that's considering each step, including the raw materials that we're using to make the materials, the production process, using the film itself as an interactive display for retailers, consumers, and also end of life. At end of life, should then be composted, and we can then make more soil and grow more food, ultimately. So that was a fast breeze through 5 projects. And this body of work, it's varied in terms of its sort of domain and research area, but I see it as a continuum of this body of work of molecular design interactions, where we're using and thinking about the chemical and biological information that's around us and building bridges between humans and this abstract information landscape. Thank you. And if you would like to learn more about what I'm doing or keep tabs, this is my contact information. Thank you.
Wow. No, okay. Can you just spend 2 or 3 minutes to tell us how you got here? I mean, I know you did research at Samsung and JPL, but I don't know what kind of research.
Yeah. So as I kind of mentioned earlier, my journey has been a bit circuitous. I started out as a human robot and human robot interaction researcher at NASA JPL. And then I did grad school at MIT at the Media Lab. And that's sort of I went pretty deep into HCI, and that's how I got into materials and biology, biomaterials. And then after that, I think I've been walking this fine line between these 2 worlds of human computer interfaces, which is the whole world of electronics and computer science, and this realm of chemistry, biology, and material science. And now, in terms of the company, it's like in the last 6 years, my primary work has been doing materials research and working with a lot less around the work around sort of traditional interfaces. But in the field of human computer interaction, the main interest, right, is to create systems that allow humans to interact with these, of like complex systems, computer systems. And now the field has actually moved closer and closer because of wearable technology, because of health data, because of trying to interface with the environment, more and more closer into chemistry and biology. And so I think my journey is largely influenced by the gaps in the field and the interest in the field, but also my personal fascination with like this body of this whole realm of information that we don't really think that much about or interact with on a day-to-day, especially because it's so critical to the way that we live, and so critical to our health, so critical to the environment, that we do have access to that. Right. Yeah.
Okay.